So I was just mentioning, so the software has two main components, automated workflow and the visualizer. The automated workflow is the part that does pretty much all the heavy lifting is for the data processing. We've embedded therefore a Python environment using toolkits like ScanPy and SquidPy or other like spatial DE, other toolkits that are available. And it does all the things like spatial analysis, clustering, data filtering, quality control, gene correlation, pretty much everything you would usually consult a mathematician to do your downstream analysis. And that's all done automatically by the software for you. On the other side, the part that is more of interest for everyone, I guess, is the visualizer, because this is where you actually get to play with the data, to explore your data and visualize and interact with it. So you can, for spatial gene expression, you can do things like 3D mapping. And I'm going to show you in a couple of slides what these features are. But I just wanted to show you how easy it actually is to do the analysis with using VR-Omics. So in the next, I'll show you a quick video. In the video, you can see how we do a full-on analysis on a Visium, which is so Visium is a spatial transcriptomes technology commercially available. So you can see here, you just pretty much really select the folder as you get it from the vendor and load it in, and then you just fill out pretty much this one page. So you kind of set in your filter parameters, you decide if you want to create the output plots and do spatial or other genes analysis, and you click process, and that's really it. So it takes you roughly, I don't know, like 10 seconds. And on your local machine, there will be folder generated for each one of your samples, where you get like all your figures, all your spatial plots, histograms, UMaps, and so on, but also all the files that are necessary for you for the analysis, downstream analysis, like the HDF5 files, CSV, count matrix, and so on. And the cool thing is that these folders are fully compatible to VR-Omics, so you can take them and can put them back into VR-Omics to visualize them. And to show you how easy that is as well, I'm going to show you in this video. So now we're just going to select the folder that was created from our local machine. Then you get like this little overview, and then it enters the visualizer, where you can now have a fully interactive data set there, where you can, for example, here like look at the cluster information. But you can also do other cool things, which I'm going to show you throughout the presentation. One very important thing that we're really passionate about is actually three-dimensional data analysis, which means spatial data doesn't only exist in a two-dimensional space, but obviously, if you, for example, think about the heart, it goes without saying that it's a three-dimensional structure. And if you want to do spatial transcriptomics on that, you have to pretty much take it, slice it down to multiple sections, do spatial transcriptomics on each one of them. And this is something that Asbjørn All did in 2019, which was published in Cell, that took the human developing heart in 6.5 weeks, did 9 slides, and created a 3D data set out of it. The problem was only, it took them a lot of software developers and bioinformaticians and whatnot and manpower to actually present the data in the long run, and also to make it interactive and to really work with the data. And this is not feasible, and it's not really how we should do it if we think about having a lot of experiments running in three-dimensional space. Therefore, VR-Omics allows you really easily to create 3D data sets by just uploading multiple slides. And just to show you, this is how we would do that. We would just select multiple slides. Let's say these are 5 slides that belong together in one 3D data set. You get here a little overview of all the slides that you are selecting, and this here is an alignment tool. So now you can like select the rotation to the slides each other, or like the distances to create your 3D data set. And that's what you get, like a fully interactive three-dimensional data set made from these slides. And there you can clearly see, for example, like the overlaid it with a three-dimensional mesh, which you can also do if you have one available. And then you can see like clearly, like, for example, these beautiful cluster regions within the chambers of the heart. And this is how three-dimensional data is meant to be analyzed in one big thing to see the whole big picture. Just to go through a couple of other features in the that you can use. For example, one of the things clearly would be you want to look for your favorite gene. For example, here for the Merfish mouse brain data, if you search for the gene OPRM1 here, it's pretty much just selected and will be shown on the slide. For example, here you can clearly see like how it's expressed very highly, like in the left and right regions. Then you can also apply a threshold, as I did here, just get rid of the noise and make it a bit more clearer to see. Additionally, you can also look at your genes in a binary manner, which means it will only show all the locations where the gene is expressed. For example, here in the Xenium breast cancer data set, selecting the gene, and you only see the region where the gene is actually expressed in. Another cool thing is for comparison, you can search for 2 different genes at the same time. So for example, here for Visium data, so now we're looking for one gene, switching it to binary mode to see where the gene is expressed. Now we select the second gene and just apply it. And now we can see within one data set, green is the first gene we selected, red is the second gene, and white is all the regions where the genes are same expressed. Additionally, we have features the side-by-side comparison that allows you to pretty much create a duplicate of your slide and look at two different gene expressions at the same time. So you can really like visually compare two different gene expression patterns. As you can see here, now we have two different gene expressions on each slide. And then you can also merge them back together into one slide, which happens to have like a vector-based difference analysis between each one of the spots. And it will show you within one slide how different both the gene expressions are to each other. Of course, you also want to select regions of interest. You pretty much just draw on the slides. You can export these regions. They will also be transferred to the side-by-side comparison. And of course, you can do the same thing in 3D as well. So we have an algorithm in place that detects underlying locations in multiple slides and creates a big three-dimensional region of interest out of it. And you can export the whole thing and create sub-regions of your data set. So in the beginning, I told you that we have virtual reality capabilities as well. At any given point, really, you can use the software as a virtual reality environment if you want to. So you just pop on the headset and press the button, and it will automatically switch into VR mode, which means everything I showed you is also available in VR, but just to show you how it look like. Here's the data set from the beginning, just as a VR environment now, which might be really helpful if you really want to dig in and you want to interact with the data a bit more into detail. And you can also do things like customization of the whole for example, like how the locations are shown. You can switch into like spheres, or you can change the size and transparencies and stuff like that. So I can't cover all the features. Another thing would be, for example, UMAP feature, TSME, like all of this is available. And just to show some of the other data sets that we show and the for the other methods that we also use, like also nanostring, TomoSeq, like there are a lot of different spatial transcriptomics methods available. And we try to expand it as quicker or like as good as we can. And with the last couple of minutes or quickly, I just want to go through the experimental design. So because at this stage we have the software, but we kind of really lacking the three-dimensional data for it because not many experiments are run. Like they're kind of starting now, but like we don't really have the data to analyze data often in 3D. Therefore, and since we are located in the Royal Children's Hospital, we are really curious about finding out a lot of genetic disease as well and how they are caused. And specifically one that we are interested in is a disease called dilated cardiomyopathy, which pretty much has means that like usually the left ventricle of your heart is kind of enlarged, and that weakens the heart function of your heart, makes it hard to pump blood through the body. This can have genetic mutation as cause, but can also have viral infections, alcohol abuse, or other factors. The symptoms are usually fatigue, shortness of breath, can lead to death even. So we're really curious to see like what's actually going on within the left ventricle of the heart, or pretty much throughout the whole heart, and how this disease is developed. To do so, we're working with a mouse model, and the plan we're having is we want to create in total 4 three-dimensional data sets of like 2 different time points at 4 and 21 weeks, where we compare a wild type with DCM, whereas we chose the 4 weeks because this is really where it starts, the disease starts to develop. And in 21 weeks, we have a clear phenotype where we can clearly tell like the heart is already, the left ventricle is very enlarged already, and the heart in general also has like a very large size. To do so, we will again slide multiple sections from each of the hearts, whereas we want to try to aim to at least have 10 to 15 slides, if not even more. And then additionally, we want to use BGI's seq technique, which has higher resolution than the one that I just presented with the human developing heart. And then I've shown you that there was a 3D mesh overlaid with the data set that we had that I showed you at the beginning. As you can see, the 3D mesh is like really low resolution, and they generated from multiple sections. We actually try to aim to have higher resolution by using micro CT instead. So we will create 4 hard meshes to have like local orientation of the data when we overlay it. And this here is just like for representation, it's the original data set, but it would look similar-ish like this. So in total, we would gain 4 spatial temporal atlases of the different time points where we really can identify the molecular drivers and underlying the cardiomyopathy progression, but also like explore gene regulation during heart development, but also disease development. So that's it from my end. If you want to find out more, use the QR code to scan. And I just quickly want to thank the whole group, the heart regeneration group, the Schreiber group, who's our immersive analytics specialist, the MCRI, and Novo Nordisk Foundation. Thank you.